Okay, um, so yesterday I gave you this brief history of neuroscience, cherry-picked by myself. I mean, the things I like most. And today I'll jump directly into this topic of brain criticality. And the subtitle, The Rise of Directed Percolation. I hope I'll make this clear throughout the, the lecture. And I mentioned the rise here because essentially I'll tell you a successful story of things that it's bad. I don't know what I can do. It's on. Is there another one? Oh. But can I control it here? Oh, okay. Sorry. Like this? Is this better? Okay, I'll hold it like this for a while. So, I mentioned the rise of direct manipulation because today I'll tell a success story of a model that works and explains some data, so it's nice. And very few go home happy. And then tomorrow I'll destroy everything, okay? With the fall of direct percolation, and many things go wrong, and we fail to understand things, and it gets messy. So tomorrow's going to be messy. Warning, this is a spoiler alert for tomorrow. I hope today is not. Yesterday I compared those two, physics and neuroscience. I like this metaphor. And so eventually, by the fifth lecture, I'll get much closer to Bill Bialec's ideas from yesterday, talking about the normalization group and modern techniques to talk about phase transitions. Today, I'll talk about brain criticality in the way it historically emerged in these ideas, okay? So I'll jump directly into this experiment, which became very famous in this subfield with John Baggs and Dietmar Plein's experiment at the NIH. John Baggs is a physicist, was then a postdoc of Dietmar Plein, who's a neuroscientist. And 2003, the Hubel and Wiesel experiment that I mentioned yesterday was in the late 60s or 70s or something. And this is 2003. So notice this experiment here. You have electrodes at the bottom of a Petri dish, 60 electrodes. So we're not yet in today's scenario that Bill just showed you a couple of half an hour ago with those silicon probes and those thousands of electrodes. Here you have tens of electrodes. And what they did, they put on top of this electrode a slice of a rat brain. Okay? So it's an in vitro experiment. It's not a live animal. It's a piece of brain which is kept alive by nutrients in this Petri dish. This is the scale, 1 millimeter here. So you have a couple of tens or hundreds of microns here across the electrodes. And what they're measuring is a type of electric activity called local field potentials. So they're not measuring the spikes of single neurons. They're measuring what this one electrode here is measuring is like the coordinated activity of many neurons around it. Okay, thousands of neurons around it. So it's a kind of average electrical activity around an electrode coming from many neurons. And this is spontaneous activity. As what else could it be? It's just a slice of cortex, right? It doesn't do anything. It's just sitting there. And what they observe is this kind of silence. Here you have the number 60 electrodes, and then time here in second. And you have this relative silence normalized by some average value or something like this. And then some kind of apparently synchronous activity, like many electrodes like light up together, and then they go silent again, and then they light up to some extent together again here, and then another silence, etc. When you look in here, you have a longer time series of 1 minute, and again, it looks very synchronized. So they put a dot here whenever this activity goes above, I don't know, 3 standard deviations or 2 standard deviations, I don't remember, above the mean, right? When you look closer on a shorter time scale here of a couple of milliseconds, you see that they're not actually synchronized. You have here this silent as defined as something below this threshold, and then ta 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 ta, and then silence again. So it's like a spatial temporal pattern followed by silence. And this event here, they called an avalanche, a neuronal avalanche, in the sense that you have a separation of time scales. The sense that the duration of this thing here, a couple of milliseconds, is much shorter than the interval between them, like in a regular avalanche, as if Brazilians knew what there was, right? We don't have avalanches, but supposedly snow takes a long time to accumulate, and then, I mean, dissipation of energy is much faster than accumulation. Right, so we have these events, and you collect them for hours, many hours. Have, I don't know, tens of thousands of these events. You can do some statistics. So they define, for instance, the simplest measurement. This is the size of an avalanche. The size S of an avalanche is the number of active electrodes in each of these events. And you can ask questions about, okay, how, what is the typical size, etc. So what they did is they collect the probability distribution of the size of these events, right, in this experiment. And they got this probability as a function of number of electrodes. It's a log scale. It's a beautiful power laws. Of course, you have a cutoff here. You have a finite number of electrodes, and they did many different tests, like taking half the slice one part of this electrodes, I'm sorry, and discarding electrodes and seeing that this cutoff of the power law changed accordingly. They also tried changing the space between the electrodes. They also tried changing the definition of the size. Instead of counting the number of above threshold events, just summing up the peak of the voltage. It doesn't really matter. I mean, however you define this thing, you obtain this power law with a 3/2 exponent. And depending on the audience that I'm addressing, the fact that this is a power law, it deserves a different so what question, right? In a physics audience, I don't have to be to take as long as I do in biology audiences to explain that this is kind of unusual distribution. It's not so, you don't find power laws that easily. This is a fat tail distribution in the sense that questions like mean size of an avalanche, typical size of an avalanche, mean plus or minus standard deviation kinds of questions are kind of meaningless because this has a fat tail. If you calculate in your head the variance, if it were infinite for the whole period, right? An infinitely distributed power law like this, the variance will be infinite, right? So it's a problem governed by fluctuations. It's a system governed by fluctuations. And you have to think how they were thinking back in 2003. I mean, if you're trying to understand how the brain processes things, the fact that in the absence of any input, just a slice, behaves like this and is ruled by fluctuations all the time, this should have a bearing on how you understand how the brain works. This is how one way to put it. So you can use jargon to impress your friends and call this non-trivial scaling variance statistics. So scaling variance is a signature in physics of many things. And I always like to remind people what scaling variance can be. So there's a couple of nice slides to remind you what scaling invariance is. One classical example comes from Didier Sornet's book, Critical Phenomena in Natural Sciences, in which he shows two pictures of two typical sedimentary layers. It's a piece of rock, right? And the question to you is, what is the size of these pictures? I mean, from here to here. I mean, or to put it another way, the way he puts it in his book. What is the, I mean, in comparison, what would be the size of a camera lens cap? I hope you guys still know what a camera is. So a lens cap is like this size, okay? If I were to put a camera lens cap on top of the wall here, what size would it be? Like this size, like this size, this size? The question is, would you risk giving me an answer? This size? The fact that nobody immediately raises their hand, that's a point. You cannot tell immediately what the scales here are, because these things, many things in nature are scale invariant, right? So he then reveals the camera lens cap on these pictures, so you have an idea. I mean, is anybody complaining about this thing? It's okay, right? I mean, could you tell if it was wrong? I mean, probably not. In fact, he's playing a prank on all of us, because look down here at the second picture. There's a second version of this picture in which now there's a person here doing like this. So this actually was a fake, but nobody complained. You cannot tell. There's no characteristic scale of this structure for it to compare with. So this is scale invariance, right? I mean, as a contrast, I don't tell you what this object is. What is the size of that? I don't know. It could be the hydrogen atom, it could be Jupiter, right? But if I do something like this, now this is 10 centimeters, because you know that humans have a characteristic size. So the size of humans is not scale invariant. They follow the Gaussian distribution with some limited width. You will not find a 7-meter tall human. You will not, ever. I don't know, right? Actually, you don't have to play tricks like this. You can just take one picture from my vacations last winter. I highly recommend this place. You haven't been there. North of Minas Gerais State in Brazil, Piruaçu National Park. And I'm not playing any tricks, no camera lens cap, anything. People who have seen this picture before should not answer the question. Do you risk telling what size is this? Is this a picture taken from a very small hole, a large cave, a very large cave, a medium-sized cave? See, there are trees, but trees also come on all sides. You cannot tell the distance very well. There are rocks here. It could be small rocks. This could be the size of my boot. I don't know. Do you risk? Can you guess what is the size of the height of that cave? It's, well, if you look here, again, I just noticed later there were some people here. Right? These are three people here. I'm sorry for the resolution of the picture. So this is a 110-meter high cave, the largest cave in this park. Beautiful place, you should go there. Ask me how, okay? Anyway, or the same picture, or this is the same cave taken from outside. You see, you have some trees nearby, but they're nearby. It's very hard to tell the size of things, right? That's the point of scale invariance. Actually, here, are some small people, again, here. That's a giant thing. Anyway, I think I have made it clear that when you cannot tell the size of, that's the sign of scaling invariance. So here, what this is saying is that you don't have a typical size of an avalanche. They come in all sizes. And if you look at the duration of the distribution, it's much uglier distribution. Again, it's only 6 electrodes. It's a power law for less than a decade. You can argue it's not even a power law, but anyway, this exponent here is 2. And when physicists saw those 2 exponents, they raised their ear like this. Oh, I recognize that. Before I go there, just let me show you that later, so they went from slices to anesthetized rats. So here's an intact animal, but anesthetized. And later on, from Miguel Nicolelli's lab in Duke University, they got data from monkeys. So these are anesthetized but resting, and you're measuring with electrodes implanted in their premotor cortex. So the animals are doing nothing. And you keep getting these power laws with similar exponents, more or less. So in the late 80s, early 90s, I was a child, I don't know. There was this surge in physics of self-organized criticality, trying to identify natural phenomena which somehow seem to converge to a critical state in which, the result of which, I'm going to connect those things, was these power laws. So forest fires in Italy or something, they don't have a characteristic size. Or rice piles, based on the idea of sand piles. The idea is that you put some grains of rice or sand here and here and here, so you have this pile which grows. Eventually, it reaches some critical slope, at which point, if you add something else, you will create an avalanche. Then it will relax. Then it will slightly diminish. And then it will accumulate again, and an avalanche. So you measure the size of this avalanche, you do the distribution, and you obtain power laws. So the system self-organizes into this critical state. That was the idea. Originally proposed by a guy called Perbach, who wrote a book with a very modest name called How Nature Works. And it's a small book. Solar flares. I like this one because it's 1, 2, 3, 4, about 4 decades. You don't control the sun, right? It's just causing these flares and with this power law distribution. So with Dietmar Planck's, Beggs and Planck's experiment in 2003, this fueled this critical brain hypothesis, which is, roughly speaking, the idea that the brain's dynamical system operates near a critical point. So this is a cartoon of a second-order phase transition. This is an order parameter. This is a control parameter. And the second-order phase transition, you change the control parameter, and nothing happens in one side of the transition. And then you reach some critical point above which there's a qualitative change in whatever you're looking at. So depending on the system, you have a different control parameter. I don't know. I will show you in a minute. Temperature as a classical control parameter in all your textbook examples you study those things, and some order parameter. Now that depends. What this idea put forward by Landau, you characterize something, you come up with something that characterizes order in your system, usually based on the symmetries of the system, etc. And then this happens. So this idea here came up initially, perhaps with Perbach and Antichiavo. They refer back to Turing, but I went back to reading Turing. I think it's a far-fetched connection. But Turing mentioned something about supercritical minds and supercritical minds, something like this, having ideas. And this is an outdated list of reviews about the subject. I have more recent ones. Of course, the question here would be what control parameters are, what other parameters are for the brain, and if the brain is critical, what is the phase transition? Let me, for those who never seen a secondary phase transition, the classical example is ferromagnetism, in which you have spins up and down, like Bill drew here yesterday, right? You remember here? And so magnetization is a control, the order parameter. You just take the average of spins up and down. If you have a surplus of ups, it goes up. If you have surplus of downs, it goes here. So if temperature is high enough, they don't get to align and have this messy behavior here, right? It's just chaos, just a mess. If temperature is low enough, you have these microscopic domains in which have some local order here, spins up or spins down. And right at a critical temperature, you have the transition between this and that. And let me show you an example, just another movie. This is a magnet. You take another magnet, you heat it above the critical temperature, so it's not a magnet. Now it will cool down. As it cools, it goes to the critical temperature and becomes a magnet. And you can see that it has crossed this limit here. Sorry. I woke some people up. Okay, sorry. Yeah, so the question here, of course, is why on earth, why on earth would you expect this hypothesis to be true? Why would people even consider that in the first place? So this has a fragmented history there, but the idea is that you can play with toy models or more sophisticated models, different kinds of models, and in those different models, theory in these models predict that you could have optimal transmission capacity if you are here at the critical point. Some optimal information processing. There's a very nice paper in neurocomputation showing this kind of thing. The largest repertoire of metastable states, so that could serve, for instance, as some kind of short-term memory as they survive dynamically in your network. And optimal sensitivity and dynamic range, as I showed with Osamu Kinouchi, which is what I'm going to talk about today, as a simple example of why a critical system could be optimal in a given way. So the idea is that, think of that as some kind of working hypothesis worth investigating to see if it holds, to see if it gives you something interesting, okay? I mean, those things seem to be nice, so if I had a brain, I would like to have those things. And if they are nice and useful, then there will be evolutionary pressure to bring you there. Therefore, that's why you observe it. More or less, that's the picture. Again, with this question, what would the phase transition then? As it turns out, so in a phase transition, so one slide crash course of phase transitions. So you have this non-trivial scaling variant behavior characterized by critical exponents. So the way this order parameter departs from the critical point is controlled by some exponent beta. And if you are sitting at the critical point and you stimulate somehow to have an external field of some sort, the way it responds here is different from the way it responds here. So here you have some kind of nonlinear response to some field H, some external field H, some external stimulus. Typically you have some autocorrelation time decaying exponentially, but then the way the characteristic time Tc increases as you approach the critical point is also controlled by some exponent. Just to give you some examples of critical exponents, And there are many more. If you can have somehow define what an avalanche is for a system, then the size of an avalanche is controlled by this exponent tau. The duration of the avalanche is controlled by exponent tau t. And then there comes the context of universality class. Two different models belong to the same universality class if they have the same exponents, even if they're different. The reason for that is the drawing that Bill Bialek made here, again, the normalization group, in which you have relevant operators and irrelevant operators. You can have two different models which are different by irrelevant things. So they belong to the same universality class because they will behave the same way near the critical point. Okay. Given the experimental results that Baggs planes obtained back then, like I said, physicists identified those exponents because they were known for a very, very, very long time, for over a century then. They were the exponents of a branching process or a contact process or what is the name of I forgot the name of the model again, 19th century. I'll remember in a minute, I'm sorry. In the jargon of the field, mean field directed percolation. So essentially, let me describe the very simple model of toy neurons. So imagine you have neurons connected with synapses, and let's imagine we take all the information about synapses, which are very complicated, and simplified, and just make it a probability, right? So this guy spiking here, it may or may not transmit the spike to this guy, etc., etc. Okay? And if the probabilities are small, this is what's going to happen. You start with some population activity, and then it will fade away. It will die out eventually. And you get to silence, and nothing happens. Right? So this control parameter here is more or less this probability here. And this is a phase called supercritical phase of this kind of model in which you go to what is called in the jargon of non-equilibrium statistical physics, an absorbing state, silence, death, nothing happens, activity stops, which is more or less what we will observe in experimental data in those silences. Now, if probability, on the other hand, is high, large enough, then you start something here, and then it is able to and excite 3 other neurons, which will then excite other neurons. So activity grows. But neurons have some refractory period. Remember, they cannot spike all the time. They have to stay quiet for a while. This is what limits the activity here. Then eventually you reach some stationary state, some constant firing rate, average firing rate in a population. So this is an active state. Neurons are firing. They're not dead. They're not silent. This is silence. This is not silence. And that's the phase transition. Here, you are in between dying and not dying, collectively, as a network. Okay? So, that's a phase transition between absorbing and active phases in neurons which are connected more or less randomly or all-to-all without some kind of spatial structure. Okay? This is in the mean field kind of limit. This is another, I'm going to show you the model in a minute, but this is a density of active sites as a function of time. We start with some subcritical value of the coupling parameter of this probability, more or less. It goes to 0. These are 3 runs, 0, 0, 0. It goes to 0. And if it's supercritical, with the same initial conditions, you stay more or less stable. And if you are in the critical point, you eventually go to 0, but you don't have you have no longer a characteristic time. The time it takes to die here is also power law distributed. You are on the verge of dying and not dying, right? Well, for that kind of model, for this kind of random topologies, the exponents are 3/2 and 2 for the size distribution and for the relation distribution, which is precisely, not precisely, but more or less what they measured in the experiments. So since despite being a very, very, very rude approximation for neuronal dynamics, it kind of makes sense, more or less. So it got people excited about it. So people read those experimental signatures as a sign that you were sitting here, because only here you observe these things here. If you are here, you get exponentially distributed avalanches. Here, in the supercritical phase, you cannot even define an avalanche because there's no silence between them. It never dies. It stays there forever. Right? And for the same universality class, you also get this third exponent that I mentioned before, is how you respond to a stimulus at the critical point with exponent 1/2. These exponents are not independent. They are connected to one another. We don't need to go into that. But so this is mean field directed percolation in a snapshot. Is that okay? Yeah? The idea of the model? It's a very simple model. I'll call it MFDP for short. So what I worked with Azumi Kinoshi was this very simple model of a generic sensory system. So, sociology of science, right? We call it. You have to be very careful when you talk to biologists. There's no such a thing as a generic sensory system. It doesn't exist. Smell, olfaction, taste, touch, hearing, proprioception, right? So, in a sense, I'm going to show a very simple model of nothing, of some kind of toy generic system. But anyway, so bear with me with that. So, do you remember the Hodgkin-Huxley spike, which takes 4 differential equations? So it goes up, then goes down, becomes refractory, and then it goes back and becomes quiescent again. So I'll replace this by this very simple cellular automaton, which is 0 here, and it spikes only if it receives some kind of stimulus, and then it becomes refractory deterministically through here, and then cyclically through here. This is called the Greenberg-Hastings cellular automaton. It's very old. We didn't invent it. And I'm going to put one of these guys in each of those neurons and build a network of neurons with these dynamics. Okay? So how do I connect them? With probabilities. So I have neuron I here, which has ki equals 4 neighbors, and I connect it with probabilities. It could be bidirectional, it could be unidirectional, it doesn't really matter. Okay? It doesn't really matter. So this is 0.1, 0.9, 0.2, 0.5. So if you sum those things, it gives you 1.7. So this is the local branching ratio of this guy. It means that if this guy spikes, goes from here to here, and these guys are available to spike, I mean, if they are quiescent, on average, you'll generate 1.7 spikes downstream, on average, right? Okay? So imagine this was an olfactory sensory neuron in your nose, and you're looking at some neuron, like Bill said, you have hundreds, I think, on the order of maybe thousands of different receptors in your nose, and then another one comes and binds. If it has affinity, it will bind to your receptor, and this neuron spikes. Now, suppose this is one of those neurons which has affinity with, I don't know, vinegar, and vinegar comes to the air and binds here. So this guy spikes. Now, it will transmit information to the other guy. This guy becomes refractory, and then refractory. This guy spikes, and then this guy comes back to quiescent. This guy is refractory. End of story. Another molecule comes in, binds, and then something different happens here because it's probabilistic, and you have these two guys spiking, and then they go refractory, and etc. Now I do this again, and again, and you got the picture. Okay? It's extremely simple model. Right. So this is the model, okay? Because now I'm going to put all this together and argue in which sense, for this very simple model, criticality would be a good thing for that system understood as a sensory system, okay? All the simulations that I'm going to show you, the results I'm going to show you is for a random network, just because in a random network, I can do calculations, then they work nicely. They're simple, field calculations, but this is not important here, okay? I'm going to show results for this kind of network, even though it doesn't matter, it doesn't have to be that network. But since showing movies of this thing is not so informative, I will rather show you movies in 2D. So here I'm showing you 50 by 50, that's 2,500 neurons. Each square is a neuron. And they are quiescent. They are doing nothing, right? Now, in these two plots are the same plot, one in log-log scale and the other in a linear log scale. On the horizontal axis, I have the intensity of the stimulus in log scale, which would be, say, something like the concentration of acetic acid, I think, molecules in the air, which comes from vinegar, which will bind to my nose, right? And the vertical axis, I have the average activity that I would measure in this network. Is that okay? So, to start with, these neurons are not coupled at all. It's a very boring model, individual neurons. And this serves the purpose of having you an example of what it means to have weak, medium, moderate, and high stimulus. So weak stimulus is this. It's like it's raining here, and then neurons spike. Boring. Stronger stimulus. This happens more often. So for each input that comes in, some neuron spikes. If it rains twice as strong, they have twice more activity. So this response, even though you cannot see because it's a log-log plot, this linear plot is exponent 1. It's a linear response. Eventually, if you stimulate too much, the refractory period of the neurons will make this curve bend and reach some saturation. And this is what you get here, a very strong stimulus. Notice that initially they seem synchronized, right? Just because they start at the same time. So there's a lot of talk about synchronization in neuroscience that somehow sometimes you get spurious results just by initial conditions, sometimes. Okay. So this is boring. Now let's add some coupling. Some very weak coupling between nearest neighbor neurons here. I'll go back to the weak stimulus, the same as before, so please notice the difference. Notice what happens now. Now, whenever I stimulate some neuron, it creates some kind of evanescent wave, excitable wave, which dies after some time, some space. They're not deterministic, they die. But what this does is to increase the overall activity that's going on in the network, that then will be sent to deeper areas of your brain that could then be interpreted by you as something like vinegar odor intensity, for instance, right? So instead of generating one spike per incoming stimulus, you generate 1 point something spikes per incoming stimulus. So you increase your response here due to coupling. This presumably is good because now if 10 to the minus 4 here, I don't know, spikes per milliseconds or whatever, was too little for you downstream your brain, maybe this jump here would increase the success of your detection, of your interpretation. So excitable waves mean amplification here in your system. Presumably a good thing. Okay, if it's amplification a good thing, well, let's have some more. So if I increase the coupling, what happens that, well, the size of these mini waves will increase, and I have more amplification. I have more signal to detect. That's good. So let's keep increasing. The more, the better, right? Well, not quite. I mean, eventually, if you increase the coupling between the neurons, eventually you'll reach a critical point. So right at the critical point, I'm on the verge of this stimulus generating a self-sustained activity or not. So I'm using very weak stimulus. I think it's time, I think 17 milliseconds or something. There you go. You create something here that is propagating. It looks like it's going to die. You're not quite sure. It's lasting a long time. So this qualitatively different behavior at criticality is what gives rise to the change in the exponent here with which you are responding. Remember, for the mean field connectivity, not for 2D, but for the mean field connectivity, this is 0.5. You have a change from one, this is 1, 1, 1, 1, 1, 0.5, 1/2. And I will argue in a minute, qualitatively, that this amounts to optimal amplification. In which sense? In the sense that suppose I increase coupling even more. Well, then I reach supercritical coupling. What does that mean? Suppose I'm here at very, very weak stimulus. This will amount to someone at the back of the room opening a bottle of vinegar, and I'll wait here until some molecule by chance will hit my nose. Very, well, for this model of a nose, look what happens if this molecule hits my nose. I'm a supercritical regime. I'm amplifying everything. That means I will smell vinegar forever. The guy closes the bottle, goes away, I smell vinegar forever. It was very sensitive, but a very bad nose. Right? In particular, I cannot detect this difference from here to here. And I'll use that in a minute. So supercritical excitable waves amount to too much amplification. I like to, okay, fortunately for you, I've used too many loud sounds already in my yesterday, today, but I have an acoustic analog for this, which is, imagine I were to tap here. Can you hear? If I were to tap very softly, you wouldn't be able to hear, perhaps, or you have hard time hearing. If I now were approaching the microphone to the loudspeaker, fortunately I can do this close enough, there will be some kind of reverberation here. So this is a mean field analog of a connection between the sensor and itself, between the neurons, right? So, but if I were tapping always with the same intensity here, but if I were approaching, therefore increasing the coupling here, it will increase the probability that you'll be able to hear this soft tapping, right? Agree with me? Now, fortunately for you, the loudspeaker is up there, but if you were down here, I could bring it so close to the loudspeaker that what will happen? Audio feedback, right? Microfonia in Portuguese. Pee, which is the sound going like berserk like this. And then if I were tapping here, you wouldn't hear anything because you would hear only the audio feedback. Right? That's how bad this sensor is. The supercritical network. So I'm using this topology here. So I have this family of response curves. This is a collective activity as a function of stimulus intensity. And here I'm just showing this little dots, which are simulations, and the lines are calculations I can do. So it's nice you can do calculations, okay? But I won't do them here. Unless you really want me to, I think I can, but let's not do this. In the inset is the zero input, which is not here because it's in infinite, it's the log scale, as a function of the coupling, and coupling is here in this other plot, right? This is the classical cartoon of a second order phase transition, and this is the response, family of response curves. So at the critical point, this m exponent is 1/2. Like I mentioned before, that's the critical exponent for this universality class. And very interestingly, it agrees with something that Bill mentioned earlier, psychophysical responses. So this is a classical psychophysical experiment from the early the 20th century. So you stay in a dark room, someone shines a light, it's a dot of light, whose intensity is well known. So they know the luminance of the signal they are offering your eyes. And you, as a subject, report the intensity of the signal, what it looks like to you. How bright does it feel? That's why it's called psychophysics. This is physics, this is psychological response. It's a response that you estimate yourself, and this is measured physically. And then you do this over several subjects. You normalize over some maximum. You put it to 100, and do the average. What you obtain. For brightness is 0.5, for small 0.6, less than 1 exponents. Very close to what you obtain in the model at the critical point. So notice that. So I have a model that explains this experimental result, but only if I put the model at the critical point. So in physics, that's perhaps cheating, because you have to fine-tune. You have infinite values of sigma here, the coupling parameter, but it only works here. Well, but it's biology, right? So if there were a good reason to be here, then natural selection would make it happen. So far, there's no reason to be there, except for me to explain this data and convince you that the model explains that. Well, it explains that only in a single point. You take now not psychophysical data, but neuroscientific data. So this again, linear log response of retinal ganglion cells of the mouse, I think. So this is how you measure intensity here. Isomerized rhodopsin molecules per rod per second. And this is a linear log scale, which is not very clear in order to observe a power law, but if you take the data, or ask your student to take the data and replot it in a log scale, you get some exponent which looks more or less, it's close to 0.5. So again, these, I'm not an expert in vision, but retinal ganglion cells seem to be already responding to intensity the same way the psychophysical responses do, right? Suggesting that the downstream regions of the brain have more important things to do than estimating intensity, right? Interestingly, you take a completely different approach. Take Antônio Carlos Roque. This is from Antônio Carlos Roque's group in Ribeirão Preto. They do hardcore computational neuroscience. So they build a model of the retina in which they take biophysical parameters of the membranes of the rods, which are here, taken from all the literature about rods in mammals that they could gather, and take all the details, the best rod model that they can build up from the literature with biophysical parameters. So there's not much room for fitting anything. You take from experimental data. And then you connect to, I forgot the names of the layers in the retina, rod bipolar layer, cone layer here, and the proportion of rods to rod bipolars is respected according to biology, and you build a detailed model of the rod bipolar cells, and according to the best fitted parameters, and a macrine cells layer, again respected the the projections and the ratios between neurons here and here, and then to the cone bipolar layer, to the ganglion cell layer, using the best ganglion cell model that you have, and you run this model. And you see what kind of response you get from a stimulus here, which only gets at in this initial layer, okay? And what you get is like exponent 1.5 again, without fitting. They cannot fit anything. They have to do everything taken from biological data. Again, of course, this is a much simpler model, but it gives you the same exponent at the critical point only. Let me jump very briefly 10 years ahead and look at this paper by Dante Calvo's group in Buenos Aires, in which they take the human connectome, put a model on top of each node, run the model, and take subcritical, supercritical, and critical dynamics, and then measure the functional connectivity that emerges from these dynamics. Meaning which nodes are connected to which, correlated to which, after we run the dynamics. And this is the experimental data here, obtained by fMRI. This is the data from the model, obtained at a critical point. Essentially, it works, the match with experimental data is best when the model is a critical point. The same year, Gustavo Deco's group in Barcelona did kind of the same thing. They had this structural connectivity matrix of the human brain, I think, and according to this DTI that I showed you before. And they put some model on the nodes. They run the model. From the model, once the model is running, they calculate functional connectivity by taking, say, correlation between the nodes. And then they compare what the model gives as a functional connectivity with the matrix of empirical functional connectivity obtained from fMRI. And their plot is a little bit better than this one, I think, because here in the vertical axis, they show the fitting between empirical and simulated functional connectivity. And here, the coupling strength, one of the parameters of this model, and the best result is obtained at some kind of critical point of the model. The highest fitting between simulation and reality and experimental functional network. Okay, but let's go back here. Again, the model only works if I'm here. So, is there a reason for me to be at the critical point? Let me argue that, yes, I think we think there could be, according to this extremely simple model. So, I'm showing again the same family of response curves, except now it's more convenient for me to use the linear log plot, okay? So you have here subcritical coupling. As you increase coupling, this low stimulus response increases. You have the critical point. Then a supercritical phase, you have this the self-sustained activity here, non-zero activity, right? And it keeps increasing as increased coupling. So coupling goes like this. So biologists and engineers, and basically everyone, have some definition of dynamic range. So for this kind of sigmoid-like plot, let me go through this definition of dynamic range. Suppose you had this response curve here, which has some self-sustained activity here, has not, and then increases and then saturates here. You have some lower saturation, some upper saturation, right? And suppose this is one of my fictional olfactory sensory neurons. And this is in your nose. You want your brain to receive information. So what your brain receives is not this thing here. What your brain receives is this thing here, right? Whatever is spiking. This is what in neuroscience is called rate code. I'm assuming that you can take some average over some time and calculate some average rate, and information is there. There's a whole book about how this is not always the case. Bill has a very book which was defining my life. I was my postdoc with Katja Lindenberg in CSD, and I didn't know what to do with my career. I took your book from the library. It's called Spikes, and it deals with information theory in the one of the neurons of the fly as it watches TV. It had a very deep impact, and thank you, Bill. You should read the book. It's amazing. Anyway, this is assuming that you can measure some averages here, okay? So take this example curve. Suppose your stimulus here was 10 to the minus 5. It gives you this response here, right? Suppose it were, now I increase the stimulus tenfold, 10 to the minus 4. What is the response? Well, the same response, right? Effectively. Not exactly the same, but almost the same. Taking into account that you have error bars here, you would not be able to tell this stimulus intensity from that stimulus intensity. So from that point of view, this curve doesn't code well this guy, this stimulus in this range here, because it's non-invertible. You cannot invert from here. You don't know if it's 10 to, if I tell you that this is your firing rate, you cannot tell me safely, accurately, whether the stimulus comes from here, or even maybe from here, right? You understand? So this is bad coding, and this is also bad coding. You have the same problem here, right? So an operational definition of a dynamic range, which biologists use, is very crude, and you could do, again, recommend going to Bill's book on information theory to measuring mutual information between two things, like stimulus and response. Here, I'm going to use something much cruder. Throw away this region. Okay, take this interval from the minimum to the maximum. Take the 10% above the minimum and throw away. And 10% below the maximum and throw away. It's just very brute force and very widely used. And whatever is left, which is here, you assume you are far away from these plateaus, and therefore you can, this is reasonably invertible, and you can use as a, you can say that here, these things here code well for these things here. And therefore, if you throw away this thing, the dynamic range definition is this intensity here divided by this intensity here. You take the log 10 of that. So this will be 2.2 decades. 2.2 decades times 10 is 10 decibels. Okay, just use some normal eight-dimensional unit. Okay, so this 22 decibels is the size of the dynamic range. So the idea is that the larger the dynamic range, the larger the span of stimulus intensities that I can operationally process. And presumably, larger dynamic ranges will be a good thing in general. I mean, again, this is a generic sensory system, right? So let's apply this idea to this set of family curves. So I'm going to assume that as I increase coupling here, this upper limit doesn't change much. So I'll keep this fixed for the sake of pedagogical reasons, okay? Let's keep this fixed. So let's start with this uncoupled neuron thing here. No coupling. So there's no F naught here. F naught is 0. So I think 10% of this guy here, which is the maximum here, is points somewhere here. And therefore, for this lower curve here, this is the point of minimum detection according to the arbitrary criterion of 10% below and above threshold. Okay? So the dynamic range for this guy is this guy. I don't know, 17 decibels, something? 18? Okay. Now, as I increase the coupling, dynamic range increases. Because why? You know why? Because now I'm amplifying signal, right? And therefore increasing my detection at the low stimulus range. If I increase coupling, this gets better and better and better and better. Cool. until I reach the critical point. Now, if I keep increasing the coupling, what happens now? I have self-sustained activity. I have this audio feedback thing going in my system, which is preventing me from detecting this guy from this guy. So I have to go above this plateau. I have to discount this plateau, and then dynamic range is not as large anymore. And if I keep increasing the coupling, bringing the microphone even closer to the loudspeaker when I already have audio feedback, it gets worse, right? And then worse, and then worse, and then worse. In such a way that if I take, as a function of the coupling, how dynamic range increases, so it goes like this, like this, like this, like this, like this, has a peak at the critical point, and then decreases, decreases, decreases. If I make this, turn this cartoon into a decent plot, by taking the model, etc., then it becomes a nice plot in which dynamic range peaks at the critical point. Precisely where it fits different experimental data. So, in a way, we have a good reason to be there at the critical point, because at least for this simple model, admittedly, very simple model, it gives you something that seems to be useful, dynamic range. Not in all cases, not in all scenarios. I know that in some cases you would like to have the shortest possible dynamic range for some specific task. I'm just giving a very theoretical vision of a generic sensory system, which, like I said before, does not exist, right? And of course, then I was a young faculty recently hired, so I explored this in a very different kind of model with different students, different topologies, different kinds of networks, blah, 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 the whole line yard. So you can do this with different flavors of models and whatnot, and it will give you essentially the same result all the time. Of course, if you go to hypercubic lattices, you have different exponents, because that's what you expect from the renormalization group. Again, dimensionality is one of the relevant ingredients of a model which determines their exponents, right? And in parallel to all that, many people were exploring not only the fact that here you obtain maximum dynamic range, but I didn't mention, well, I mentioned that here you also obtain those power laws like you saw in those early experiments. But people were also exploring synaptic mechanisms that will take your system here, so you wouldn't have to tune it. Because after all, nobody's tuning the mouse or the desensitized or the monkeys or not, right? Their brains do it by themselves. So this is a review that we wrote with Osamu Kinoshi and Renata Pazzini, in which you can have different models. We actually made a review of models that were available, and it turns out that despite the differences in notation and stuff, all of them are more or less similar. Just by using the easiest way to measure this is you have a presynaptic neuron and a postsynaptic neuron. Postsynaptic neuron. And if very simple models of short-term synaptic depression can do things like this. So if this synapse is very strong, meaning, and if this guy spikes a lot, it is depleting its reservoir of vesicles that it has here. Eventually you run out of vesicles, and the synapse will naturally become weaker. So if it's spiking too much, if sigma is too large, if you're here, then it will eventually start spiking less. And if you start spiking, this thing stops working, then eventually you will have time for this thing to go up again, because it has a natural recovery time. Then if you're here, you go this way. So I'm just describing with hands what comes out of many different models. And what you're seeing here, that no matter how, this is again model result, right? You can start this kind of model in whatever you want, and it will eventually come here and fluctuate around this critical point. Yes? In which kind of homeostasis?
If the first neuron spikes a lot, equation for homeostasis makes alone, he
Oh, you
come
mean
back.
adaptation is
Yeah,
there?
yes.
Yes. So, Osami Kinouchi with Mauricio Girardi Chapo, they had this very nice model in which you can have both. You have both an ingredient here which controls, for instance, the slope of the gain function inside this neuron, or the threshold, or both, and dynamics for the connections. And in any case, it works. So yes, it works in simple models. We're talking about simple models, right? So we were very happy with the result, but of course, it's just a toy model, right? It's just a toy model. So in 2006, there was this Medifinol conference in Mar del Plata. Have you ever guys been to Medifinol? It's one conference on nonlinear physics and statistical physics in South America. And Dietmar Plans went there with then Woody Shu, who was then his postdoc, and Thomas Peterman was also there, another postdoc of Dietmar Shu, of Dietmar Plans. And I had dinner with them, and I said, "Look, can't you guys do this experiment? I'm sorry, I'm just a theoretician. I don't do experiments. Can you stimulate your network instead of just measuring the result? And controlling the intensity of the stimulus, and then measuring whether you have a dynamic range, and then playing with the excitability of the network to see maybe if you can go supercritical or subcritical, and see what happens to the dynamic range." And this was 2006, and it was 2009, they published the results. And I was happy for about a year because I never wouldn't have imagined that some experimentalists would listen to a suggestion, let alone do the experiment, and let alone have the experiment more or less reproduce, I mean, roughly speaking, what was going on practically by theory. So the black dots, very spread out, I know. There's no drug condition for the slice, okay? The red dots are with picrotoxin, which is substance that makes the network more excitable. Okay? Box inhibition, I think. Right? Picrotoxin, any neuroscientist? Box inhibition, right? Yeah. And then here, with another drug, AP5, the NQX, I think it blocks excitation or enhances inhibition, I don't remember. And I know it's very fuzzy and messy, much of course, right? This is a simulation for 10 to the 5 neurons with perfectly identical cellular automata. This is a slice of rat brain. But anyway, I think it also makes the point of, yeah, simple models can teach you something, right? I think we go back to this theme all the time. Later on, the same group now use intact animals, though anesthetized, and measure response in somatosensory cortex while they were stimulating the rat's whiskers, like this. And it's dirtier, but it has some kind of maximum in the middle, more or less. Right, Fernanda? No? Anyway. So I was very happy. And like I said, this is a more or less a very quick story of a simple model which gives you very nice results. Is this mean field hypercolation kind of model satisfactory model of supposedly critical brain dynamics? Well, what are the pros of this model? Well, it correctly predicts the exponents of the neuronal avalanches that I showed you in a couple of experiments. Tomorrow I'll show you some other experiments. It correctly predicts some psychophysical response exponents, plus its optimal dynamic range. So it would explain, it gives you a possible mechanism for these psychophysical exponents, and also in ganglion cell response that I showed as well, and a reason why it could be that way, because then it optimizes something else. So that's why it is non-trivial, because non-triviality is useful, presumably, according to the model. And therefore, this created a useful picture. People thought a lot about this kind of model for over a decade, trying to understand different instances where these avalanches were measured. I haven't shown you here, but there are avalanches in EEG data. So it's a completely different scale. It's no longer the scale of a couple of square millimeters, but the whole surface of a brain. Then people worked on fMRI avalanches, which now is 3D. It's everything in your brain in a completely different. So each voxel is 1 cubic millimeter, and time scales is over seconds. Not, a characteristic peaks of activity is over seconds, not milliseconds. And the scenario kept pervading this literature, so to speak, right? This kind of phase transition between silence and non-silence. And what I'm going to talk tomorrow is how this theoretical idea kind of faces its limits when you choose some kind of more interesting data. Just let's let the animal do something, non-anesthetized. And things start to break down very quickly. And our theoretical capacity got shaken by that time. So I'm going to tell a story of 10 years ago. And then in the 4th and 5th lecture, come back and see how other techniques came to rescue us to have a better understanding. Okay? So I think 10 minutes early, just let me thank. Most of the work I showed today was Osami Kinoyoshi, but many people worked in other parts of the things I'm going to talk about. Thank you.
So thank you for the lecture. So I have a question about this coupling. So do you know if it's possible to assign some energy cost to maintaining the system at some specific coupling? So if you decrease a little bit the coupling between the neurons, then you spend less energy, and then it's better for the brain to be there, even though you don't encode as much information as you can. So maybe my question is whether you know if there is some trade-off between the information encoding and energy consumption.
I'm trying to remember whether in that review paper that we wrote, we included some model that took that into account. I can't remember. But people have tried that. But I think that people have tried. Remember this guy somewhere in the East Coast, Shklovsky or something, I forgot his name, who worked precisely on the modeling the wiring, and based on the energetic cost, and could predict a lot. But then again, he was not interested in this, but right in this. So yes, I think people have looked at it, but I'm not sure how to connect with that. Before I would delve into that, what you're saying, the first thing that any neuroscientist would protest immediately immediately. But that, dude, you're talking about the cortex, and there are no inhibitory neurons in your network, right? You don't do that. So you include inhibition, things get incredibly more complicated very quickly, but it can be done as well. And it was done by Mauricio Girardi Chapo here and Ozami Kinouchi. Some other where it still works. But I think I'm diverting from your question. I'm sorry. Yeah. Is it okay?
Hi. So I was wondering if maybe the model is somehow sensitive to the network size. And also, you tried this model not only on random graphs, but you showed that you tried it in like different architectures, and you found like mostly similar behavior. But I was wondering also if you found like any particular differences depending on the network architecture.
Yeah. So there are some simple results. Since you have this log of stimulus here and log of response kind of curve that I showed, you get like 1, 1, 1 for supercritical response, and you get less than 1 in the critical case. For this random graph thing, so this mean field direct propagation universality class, I told you that the exponent is 1/2, right? And the lower the exponent, the higher the response here will be, right? So here's 1/2. As it turns out, I'm not proving this, but this is like, mean field is like infinite dimension. If I go to 3D, I forgot the value of the exponent, but it's less than 1/2, right? So I think it's 0.4, I forgot the rest. It goes like this. So dynamic range is even higher. If I go 2D, it's like this. If I go 1D, it's 0.11 or something, if I remember correctly, the exponent. So if you change the effective dimensionality of your network, it will change the exponent and therefore affect the value of the dynamic range that you observe. Essentially, more structured networks lead to higher dynamic ranges. I think it's more or less safe to say that. And but of course, it depends on what question you're trying to answer. If you are a neuroscientist interested in a specific part of the brain, then details matter. For instance, this network, I'm stimulating everybody and measuring everybody. Many systems will not be like this. You have some part of the brain, some will receive stimulus, some will send it away with some preprocessing in the middle. So the fact that you're stimulating here and measuring here changes things and tends to increase dynamic range even further if you are at a critical point. So given that idea, you can play a lot with different questions, including the structure. But so to give you a very short, if you're staying in the same kind of scenario in which you stimulate everybody and measure everybody, we know that if you take, for instance, a 3D nearest neighbor network and put some a handful of random connections, turning this into a small world graph, it very quickly throws this thing into the mean field universality class. This is well known in this field. Scale-free connectivities are more complicated. And as I keep listing what I'm listing to you, like hypercubic lattices and scale-free networks, possibly your neuroscientist interlocutor will be yawning, like, tell me something that is interesting to me. Because so far, the networks do not form a 3D network. Well, maybe a small world in some small region, but I want a real network. And once you go to a real network, they can give you, now you can find it. You go online, you take some real connectome. Then things, then you can no longer predict exactly what's going to happen. And people still find it. There's a nice paper by Paulo Moretti, Miguel Munoz, Nature Communication 2013, in which they take a human connectome, put one of these models on top of it, and change the parameters. What they find is that something interesting. This order parameter here as a function of a coupling, you have the secondary phase transition. They find that in this region here below the subcritical regime, you have some kind, what is called a Griffiths phase, in which you have generic power loss with continuous vary exponents. Simulation results, and they argue why this could be the case, and then therefore you could have this kind of optimal results akin to critical points in a larger region. So it starts getting complicated once you get into the details. But again, it depends on who you're talking to. This phenomenon could be interesting for physicists, but not to a biologist, perhaps. I don't know.
So thanks a lot for the lecture, sir. Yesterday you said that the neuron itself has a kind of stochastic behavior, something like that. So when you are talking about the probability of the coupling, you are wonder more of this own behavior of the neuron, or it's more related to the strength of the coupling or the kind of synapses, something like that?
I'm projecting all my ignorance and all the things that I don't know into the single number, P, and hoping for the best.
Okay.
And trusting that it will make sense. Yeah.
Okay, thank you.
But I'll go more with the synapse because the spike of the neuron is very well behaved.
Okay, so if we don't have more questions, let's thank Mauro again for the lecture.